Hi, my name is Joel Peterson. I'm a soil biochemistry and environmental toxicology at the University of Wisconsin-Madison. And today I'm going to discuss the potential transmission of disease through soils, focusing specifically on prion diseases, such as mad cow disease and chronic wasting disease of deer and elk. In this lecture, I'd like to talk about prions in the environment, the potential role of the environment in the transmission of prion diseases, as well as waste disposal issues related to prions. First of all, prions are the causative agents in a family of diseases known as transmissible spongiform encephalopathies, or prion diseases for short. Some examples of these diseases are sheep scrapie, which has been known for nearly 300 years. Humans have coexisted with scrapie-affected sheep and have apparently suffered no adverse consequences. A second prion disease, the first that was discovered as a prion disease in humans, is kuru, which was endemic to the Foray tribe in Papua New Guinea. It was spread by ritualistic cannibalism, and the incidence of this disease has dramatically decreased with changes in dietary practices. Bovine spongiform encephalopathy, or mad cow disease, is probably the one most widely known. This disease has been transmitted in cattle populations, primarily in Great Britain, but in other parts of Europe and in North America as well, through the imposition of industrialized cannibalism, that is, feeding cow parts back to cows. And this disease is of particular concern because there's strong evidence that this disease can be transmitted to humans. Other human prion diseases include Creutzfeldt-Jakob disease, which has an incidence rate of about 1 in a million. There are a few other human prion diseases, including fatal familial insomnia. A prion disease of relevance to Wisconsin and other states within the U.S. is chronic wasting disease, which affects elk and deer. The known range of chronic wasting disease has spread dramatically in the last few years. Chronic wasting disease has been known since the late 60s and was believed to be isolated to captive and wild deer and elk populations in northern Colorado and southeastern Wyoming. This disease was present in mule deer and elk. In recent years, its known range has expanded through many of the western and midwestern states, as far west as Utah, as far south as New Mexico. We have a great concern here in Wisconsin about the spread of disease now among white-tailed deer. The disease has recently been found in Illinois and just this year in New York State and in West Virginia. This disease, as I mentioned, was first discovered in mule deer and elk, but has jumped species to white-tailed deer, and there's much concern that this disease will spread further because the density of deer in the east is much greater than the densities in western states. And now I want to shift gears a little bit and start talking about prions in the environment, why we're interested in prions in the environment, some of the information that has led us to hypothesize that soils may play a role in the transmission of prion diseases. It is clear that an environmental reservoir for prion infectivity plays a role in the transmission of at least 2 prion diseases: chronic wasting disease of deer and elk and sheep scrapie. As far back as the 1940s, people noticed that introduction of healthy sheep onto plots of land that had previously contained diseased animals resulted in the healthy flock coming down with the disease. This has been more recently demonstrated a bit more conclusively in both Iceland and the United Kingdom. Similarly, among captive elk in Colorado, researchers noted that when healthy elk were introduced to paddocks that were previously occupied by diseased animals, that the healthy elk contracted the disease despite disinfection attempts prior to their introduction. And the period between the removal of diseased animals and the introduction of the healthy animals ranged from 1 to 3 years, suggesting that the prion protein can survive in the environment for at least several years. Recently, a controlled field experiment was conducted by Mike Miller and coworkers in Colorado, in which they subjected mule deer to 3 different treatments to see if chronic wasting disease would be transmitted. In one treatment, they housed mule deer with naturally infected deer. In a second treatment, they allowed an infected deer carcass to decompose and then introduced healthy deer nearly 2 years later. And in a third treatment, they maintained diseased deer and removed them and then introduced healthy deer a little more than 2 years later. Each of these treatments was replicated 3 times. That is, each experiment was conducted in triplicate. There was also controls in which healthy mule deer were held in pens that had never had any chronic wasting disease infected animals in them. In each of the treatments, at least 1 deer became infected, and then the experiment was halted. None of the exposed deer were infected. So this controlled field experiment demonstrated a number of things. One, that the presence of carcasses on the landscape could contribute to the transmission of prion disease, that residual excreta, feces and urine, may also contribute to transmission of the disease, and significantly, none of the exposed deer were infected. These data, in conjunction with a few other lines of evidence, have suggested to us that soil may serve as a reservoir for prion infectivity in the environment. One line of evidence is that soil is ingested by both wildlife and livestock, and this can be either incidental or deliberately. Incidental ingestion occurs during feeding. For example, a sheep may pull up a tuft of grass and consume the roots and the soil associated with the roots. Soil splashed onto the leaves of bushes may be consumed by deer. Animals may consume soil while grooming. Animals may also consume soil deliberately. Herbivores may consume soil in areas that have high content of particular minerals that they are deficient in their diet. These areas are known as mineral licks. Quite a large amount of soil can be consumed at such sites. There's been some research on the amount of soil ingested by some animals, and this research was conducted in the context of trying to understand how much metals animals would consume if sewage biosolids were applied to fields. Estimates for sheep are that 4.5% of their dry matter intake consists of inorganic matter, primarily soil. For cattle, 6% of the dry matter intake may be inorganic material, and that corresponds to about 900 grams per day for a 500 kilogram cow that is pastured the entire year. Another line of evidence consistent with soil serving as a reservoir is that the abnormally folded form of the protein accumulates early in the disease in the lymphoid tissue of the gastrointestinal tract. This suggests that oral transmission is important in prion diseases, and it also suggests that prions may be excreted in feces and possibly saliva. And a final line of evidence is that there was one experiment that demonstrates that prions can survive in soil. This was an experiment conducted by Paul Brown and Carlton Gadusek at the National Institutes of Health in the early 90s. And in this experiment, they took infected material from hamster brains and placed it in the soil contained in petri dishes. One petri dish was put into the freezer and stored. A second petri dish had holes drilled in the top of it and was then placed in a flower pot and covered with soil. The third petri dish had holes drilled in the top and bottom and put in a flower pot covered with soil. The 2 flower pots were put into their garden near Washington, D.C., and left there for 3 years. After 3 years, the petri dishes were exhumed and soil washed with water, the water filtered, and then injected into the brains of hamsters to determine the amount of infectivity remaining in the soil. The same treatment was done to the control. What they found was there was a decrease in the amount of prions in the filtered soil washes, decrease of 1 to 2 orders of magnitude. In the dish with the holes drilled in the top and the bottom, there was a small amount of leaching to the soil immediately below the dish. As you can see from the amount of prions present in the 2 dishes that were left out in the garden, there's quite a bit of difference in the amount of recovered prions. These data demonstrate that prions can survive in soil for at least 3 years. Because these investigators relied on filtered water washes of soil, it is unclear what the fate of the remaining prions was. What we do not know from these data whether the prions were tightly bound to the soil so that they were not removed by a water wash, or whether they had degraded, whether soil microorganisms were able to break down the prion proteins. This experiment also had no replicates, so that we have no idea of the variability in the amount of prions present after 3 years in soil. So, how do prions get into the environment? Decomposition of infected carcasses is obviously one way that prions may enter the environment. It's possible that they could enter the environment through shedding in feces by infected animals, possibly by saliva, and maybe by urine, although this isn't very likely. Prions have not been conclusively demonstrated to be in feces, saliva, or urine to date. This is more a reflection on the insensitivity of detection methods than it is a suggestion that they aren't present in these media. There are experiments underway using animal bioassay to examine whether prions may be present in these excretions. At the University of Wisconsin-Madison, we are conducting a number of studies examining the fate of prions in natural environments, with the primary focus being on trying to understand how this disease is transmitted, with the primary focus being in understanding how chronic wasting disease is transmitted among deer in the natural environment. So we are examining the degree to which prions associate with specific soil components. We're examining the degree to which they are degraded by soil microorganisms, and also, and most importantly, the degree to which prion infectivity is maintained. We're also examining the mobility of prion proteins in soils. Our initial focus has been on the association of prion proteins with specific soil phases, that is, the sorption of prion proteins to specific minerals. We've been focusing on quartz surfaces and 2 layered silicates, montmorillonite clay and kaolinite clay. And this slide shows electron micrographs of these minerals as well as humic substances. As you can see by the scale, or these mineral particles differ substantially in their size. The quartz sand shown here, the scale bar is 100 micrometers, the kaolinite 10 micrometers, and the montmorillonite clay 1 micrometer. So we expect that the surface area available for sorption will play an important role in the sorption of prion proteins. But surface properties are likely also important. The next few slides will show you results of experiments that were conducted using immunoblots. So what you see in the slide is the result of an immunoblot experiment on X-ray film. The bands correspond to prion proteins that have been detected using antibodies. The numbers to the left of each immunoblot are the molecular masses of the molecular size markers that we run with the protein. We use centrifugation to separate bound from unbound prions. The lanes in the gel marked SUP, SUP, stand for supernatant, or the unbound fraction of prions, and the lanes marked PEL for pellet correspond to the mineral particle associated prions. In the lower left corner of each image you see MAB3F4, that just indicates the monoclonal antibody used for detection. In the left-hand immunoblot was a comparison of sorption to montmorillonite clay and quartz sand. And from this immunoblot, we see that all of the detectable prion protein is associated with the montmorillonite clay, but to the limits of our detection, none is associated with the quartz sand surfaces. This is not entirely unexpected because the amount of surface area present in the sand treatment is much less than for the montmorillonite clay. On the right-hand side is an analogous experiment comparing kaolinite and montmorillonite, and again, all or nearly all of the added prion protein is associated with the mineral particles. Another point I'd like to bring out on the left-hand panel of this slide is the apparent change in molecular size of the protein upon desorption from the montmorillonite clay. There appears to be a shift in molecular mass, and this would most likely correspond to some cleavage of the protein, which is an unusual finding and has not been reported for other proteins. We qualitatively examined the kinetics of prion sorption and cleavage by exposing prion proteins to montmorillonite clay for various time periods. And as you can see in this slide, adsorption to the clay was fairly rapid, much of the adsorption taking place within 1 minute of contact with the clay. Even at 1 minute of contact, we can begin to see some cleavage of the protein. Because we hadn't seen reports in the literature of cleavage of proteins upon desorption from clay, we were concerned that perhaps our clay samples were somehow contaminated. We therefore took a number of measures to ensure that there were no proteases present in these clay samples, that is, enzymes that would break down proteins. We did this both by adding protease inhibitors and by boiling the clay to denature any proteins that would be present in the clay sample. We also considered the possibility that some other minerals could be present in our clay samples, although we considered this unlikely. But as a precaution, we treated the clays with chemicals that would dissolve amorphous oxide coatings. Both the treatments to eliminate any potential protease activity and to remove oxide coatings had no effect on the cleavage of the protein. That is, the protein was still cleaved upon desorption from montmorillonite. We're therefore quite interested in what portion of the protein was cleaved upon desorption from the clay. In order to do this, we used antibodies that were directed against other portions of the protein. On the left-hand side of this slide is an immunoblot showing prion protein that has not been exposed to montmorillonite clay in the left-hand lane, and the right-hand lane prion protein that has been exposed to the montmorillonite clay. And we used a polyclonal antibody that was directed against the N-terminal amino acids, that is, the amino acids on the unstructured N-terminus of the protein, and found that we had immunoreactivity for the intact prion protein, but not for the protein that had been desorbed from the clay. That is, the N-terminus, or at least part of the N-terminus, was not present in the cleaved protein. On the right-hand panel, we exposed both intact prion protein and prion protein that had been exposed to proteinase K, or PK, a proteolytic enzyme that removes the N-terminus from abnormally folded prion protein, and probed with the antibody we had used in the previous immunoblots, and found that treatment with proteinase K produced the expected shift in molecular weight, but no further shift in molecular weight was apparent in the protein desorbed from the clay, suggesting that no other changes in protein size occurred. That is, the cleavage appears to be at the N-terminus and not some other portion of the protein. In all of the experiments that I've discussed up to this point, we've removed prion proteins from mineral surfaces by boiling in sodium dodecyl sulfate, SDS, to examine the strength of prion attachment to clay surfaces. We used solutions of different composition to see if we could desorb the protein. In one set of experiments, we used chaotropic agents, urea and guanidinium hydrochloride. These are chemicals that disrupt the hydrogen bonding structure of water and have been shown to remove other proteins from clay surfaces. So in the left-hand panel, you see lanes corresponding to these chaotropic agents with a 1 and a degree sign primary extraction followed by a secondary extraction using sodium dodecyl sulfate. And in these experiments, no detectable protein was removed using the chaotropic agents, but we got maximal recovery with the boiling SDS. Using solutions of different ionic strength likewise did not desorb any protein. So in summary, we have found that the association of prion protein with montmorillonite clay is extremely avid. We have to resort to boiling and denaturing detergent to remove the protein from the clay surface. This suggests that the prion protein, once attached to clay particles, is unlikely to detach from them. That is, the mobility of the protein would be limited by absorption to clay surfaces. The absorption of proteins to clay surfaces and other surfaces often modifies the function of the molecule. For example, studies with enzymes have shown that when the enzyme is absorbed to a clay surface, there is a modification of the activity of the enzyme. We are therefore curious to find out to what degree absorption to montmorillonite clay might affect the infectivity of prion proteins. So we adsorbed prion protein to montmorillonite clay and injected the prion clay complexes into the brains of weanling Syrian hamsters. In addition, we ran a number of controls, the first control being inoculating them with montmorillonite clay without any prions, the second control inoculating them with prions that are not associated with clay, and running a control to account for any prions that might not be attached to the clay particles but might co-sediment with them. Okay, this slide presents the results of these intracerebral challenges of Syrian hamsters with the different samples. The x-axis shows the time after intracerebral inoculation in days, and the longer the incubation period, the smaller amount of prion infectivity present. Shorter incubation periods correspond to higher amounts of prion infectivity. The bottom bar corresponds to montmorillonite without any clay added, and you see there is no infectivity over the course of the experiment. Surprisingly, all of these hamsters lived, even though we took no precautions to sterilize the clay. The blue bar immediately above that, marked PRP-SC pellet, is a control to account for any prion that might co-sediment with our mineral particles, and this shows again a very minute amount of infectivity, indicating that our separation procedure is effective. The bar marked PRP-SC soup is our positive control, that is, prions that have not been in contact with montmorillonite clay, and they had an average incubation period around 100 days. And finally, the top bar is prion montmorillonite complexes, and they had an incubation period of somewhat less than 100 days. And these data demonstrate that prions associated with clay complexes retain infectivity. In fact, their infectivity is not diminished by absorption to clay surfaces. And this is quite a surprising result, especially given the results of the extraction experiments that indicated that the association with the clay surfaces is quite strong. A current understanding of how prion diseases propagate requires contact between the abnormal and normal forms of the protein. Given our extraction results, it appears unlikely that the brain possesses microenvironments that would extract prions from clay surfaces. So we hypothesize that the conversion of normal protein to the abnormal form is occurring at the clay surface. We've also been conducted research in the fate of prions in engineered systems. And this is due to the large amount of potentially infected waste that is produced during the management of chronic wasting disease and mad cow disease. In the case of chronic wasting disease, states have adopted different strategies to deal with it. In some states, such as Wisconsin, large numbers of animals are harvested from infected herds, and then those carcasses need to be safely and effectively disposed of. In Wisconsin, around the Mount Horeb area, the Department of Natural Resources was intending to harvest 25,000 deer from an area 360 square miles in area. This is a large number of carcasses to dispose of. In the case of bovine spongiform encephalopathy, mad cow disease, the one case of mad cow disease discovered in the state of Washington in December of 2003 resulted in the need to dispose of 2,000 tons of potentially infected beef material that had either low levels or no infectivity. As of the recording of this lecture, that material is being held in refrigerated containers awaiting a decision by the Environmental Protection Agency as to what to do with them. So in the case of both mad cow disease and chronic wasting disease, management responses create the need for safe and effective disposal of potentially infected material. One method of disposal being considered is landfilling. This may be a preferred option because it is relatively inexpensive. In a sanitary landfill, waste material is isolated from the surrounding environment by subsurface and surface barriers. Because water infiltrating through the top of landfills and because of the decomposition of wastes, pollutants could migrate through landfills into subsurface soils and eventually to groundwater. To prevent this, modern landfills are lined with clay and or geotextiles to prevent the leaching of contaminants to groundwater. The water that leaches through landfills is collected by a leachate collection system, which then routes the leachate to a treatment system. One of the concerns with disposal of prion-infected waste in landfills is that prions might migrate through the waste mass and enter the leachate collection system. To address this concern, we have been examining the movement of prions through soils, starting with very simple systems. In order to assess prion mobility in landfills, we have been conducting a series of prion transport experiments, that is, examining the transport of prions through columns of soil in the laboratory. Prions are known to stick to a variety of materials, so we therefore tested potential materials that we could use in the construction of the columns and found that Teflon was the best material. This slide shows one of the Teflon columns that we fabricated for these experiments. The column is packed with a soil, and then we add prions to the top of the column and then allow a solution of selected composition to pass through the column, and we measure prions as they exit the column. Our initial experiments have focused on extremely clean quartz sand, so we could understand some of the fundamental processes that are controlling prion movement in soils before we work with more complicated and more realistic systems. This slide shows the results of 3 of our prion transport experiments through quartz sand. The vertical axis shows the amount of prion present in column effluent samples in units of milligrams of brain equivalent. The horizontal axis shows the number of pore volumes that have passed through the column. It could also be viewed as time. A chemical constituent that does not interact with the quartz sand would come out of the column at 1 pore volume. As you can see from this slide, the movement of prions through the quartz sand column is retarded relative to a conservative dissolved tracer. These curves are known as breakthrough curves, and for the prions, the peak of the breakthrough curve occurs between 2 and 3 pore volumes, indicating a retardation in movement. The 3 treatments shown on this slide correspond to solutions of 3 different pH values, pH 4, 7, and 9. pH 4 is below the range of isoelectric points reported, and pH 9 is above the range of isoelectric points reported. At all 3 pHs, the surface of the quartz sand particles is negatively charged. At pH 4, the bulk of the prion protein fibrils are expected to be positively charged, pH 7 and 9 negatively charged. Consistent with what we'd expect based on electrostatic interactions, more prions were retained in the pH 4 column than in the other 2 columns. But in all cases, we did not recover all of the added prions in the column effluents, indicating that some process is occurring that is irreversible in the time scale of our experiment. Although prions are transported through these columns, a fraction of them are retained. We also examined the movement of prions as a function of ionic strength, and this slide demonstrates that there's greater retention of the prions under higher ionic strength conditions and lower ionic strength conditions. Again, consistent with our expectations based on electrostatic interactions. These are really preliminary data and are just laying the groundwork for other experiments that are in progress, examining movement through real soils and movement through both synthetic municipal solid waste and actual municipal solid waste. Our initial experiments are showing that a fraction of prions can leach through quartz sand. We don't know yet whether they will leach through real soils or solid waste. But should prions enter the leachate collection system of a landfill, they would then be routed to a treatment system, and this is usually a conventional wastewater treatment plant. In a wastewater treatment plant, large particles are removed by bar screening, and in the grit chamber, wastewater is then allowed to settle to remove large settleable particles as well as to skim off floating waste. The effluent from the settling tank is then routed to an aeration tank where air is pumped into the wastewater and bacteria break down much of the waste present in the wastewater. This is known as an activated sludge process. The activated sludge is then settled in a settling tank. The effluent may then be filtered and disinfected and discharged to lakes or streams. The settled activated sludge, along with the sludge from the primary settling tank, may then be anaerobically digested, and the digested sludge, euphemistically known as biosolids, may then be applied to agricultural fields. So if prions enter wastewater treatment plants, the concern is whether they will survive the treatment process and whether they will be released into the environment through the effluent or through biosolids. To investigate this, we have been examining the partitioning of prion proteins between activated sludge and supernatant. We've been examining several different types of sludge. This slide shows Western blots for partitioning experiments between rendering sludge, designated as RH, activated sludge from a wastewater treatment plant, designated as AS, and anaerobic digester sludge, designated as AD. In these experiments, we've been using brain homogenate as a source of the prion protein rather than the enrichments that we had used in the previous experiments. And I'd like to draw your attention to the lanes that are marked AS. One is for the supernatant and one for the sludge. For the activated sludge, the bulk of the prion protein is associated with the sludge, which is what we would expect based on the known properties of prion proteins. A relatively small amount was retained in the supernatant, so this suggests that prions, should they enter a wastewater treatment plant, would most likely, one, not be biodegraded in the activated sludge process and would sorb to the sludge and be routed to an anaerobic sludge digester. So the next phase of our research is determining whether anaerobic sludge digestion results in inactivation of prion protein. I'm not going to discuss the results of the re-rendering sludge or the anaerobic digester sludge because we are still working out the method to recover prions from these types of sludge, which has proven quite difficult. So in summary, our research is demonstrating the plausibility of soil as an environmental reservoir for prion infectivity, and our work on landfills and wastewater treatment plants is beginning to provide information that will be useful to make decisions in how to dispose of prion-contaminated waste. We obviously have much to learn. This is the results I've presented are from very early stage in research. So I hope you've enjoyed learning something about how prions behave in natural and engineered environments.